talk about Darwin and Darwin's theory and some of the strengths and weaknesses of the theory. And this is going to become important because the Darwinian theory is often cited as a reason why there's no need for a God-centered view of the world. And so one can talk about, the subject is so big because it spans philosophy, it spans material science or physical science, and it spans biology. So we're wading into an ocean. And so it's extremely big topic, and I'm not going to be able to do it justice. What I am going to do is try to give you in my mind, some of the highlights. I'm going to dive a little bit deep into one subject, just as an example. Then we're going to come back out of that deep subject, and then we're going to touch on a bunch of super deep subjects and hardly do it any justice, just mention that they exist. But this could easily be, some of these slides that I'm going to show are more than a 1 hour talk, and I'm just not going to do it because there's no time. So I'm going to start with Mr. Darwin himself. In 1859, he published his magnum opus called On the Origin of Species. And in this book, he proposed basically how species evolve through a process that involved there was a number of aspects to it. But probably the two most important, fundamental aspect was that it was random mutations, that there was natural selection working on random variations. Now, mutation already has connotations of things we know now, and not being a real biologist, sometimes I will revert to language that is really neo-Darwinian, which wouldn't have been Darwin's language. So sometimes I will be sloppy. I'm going to try to correct myself. Okay, but he basically said that one can account for how the diversity of species and all this incredible amount of apparent design that we see in nature, not through any designer, but through things that are very mechanistic, very random. And it was just two principles is all we need, plus the laws of physics, and we're done. We want the laws of physics, we want random variations, and he said, well, that shouldn't be too hard to believe, because at that time, breeding of pigeons or breeding of dogs, people were very familiar with how much variation you could get. And Darwin simply said this amount of variation that you can get can be enormous, and that can account for all of life. And when he wrote this, he based it on a lot of it on his experiences at the Galapagos Islands. By the way, that I didn't know until I made this talk. This is the equator, and so the Galapagos is right at just about 90 degrees west and 0 degrees. So it's kind of a very interesting little spot, very equator-like. And so he went on the HMS Beagle, and Galapagos would like, I think it would be a very nice tourist location. Obviously, it's beautiful. And on these islands, he saw many things that he recorded in his book. And one of the most famous ones was the finches. There were these little birds, the finches. And he noticed that the islands had most dominated by one variation of each one of these kinds of finches. And some of these finches were smaller and had pointed beaks, and some of them were bigger and had kind of a rounded beaks that seemed like stronger. And he made the case that these variations are occurring because these finches are in environments that are different, and that these variations give the finch an advantage, a fitness advantage. So fitness becomes a kind of a key word here, that the finch that has the right kind of beak for that island with the nuts of the right size or toughness, or the one that was, during arid environments, the one kind of finch is able to, work better than the other, so that gives you all these variations. The theory had at that time, it wasn't like there was no biology. There were great biologists alike. But this idea that you could do this entirely through a non-designed, purely mechanistic process had a lot of attraction to it. So that like this one writer, Neil Gillespie, writes, it is sometimes said that Darwin converted the scientific world to evolution by showing them the process by which it had occurred. Yet the uneasy reservations about natural selection among Darwin's contemporaries and the widespread rejection of it from 1890s to 1930 suggests that this is too simple a view. It was more Darwin's insistence on the totally natural explanations on natural selection that won their adherence. So in other words, the author is saying that the appeal of the theory was what it was promising. It was promising a picture of life that did not involve creation. And in this case, this is Thomas Huxley. Thomas Huxley calls himself happily Darwin's bulldog. He was sort of like, he was a champion for Darwin. But in private letters, like for example to Lyell, I think he was writing this, I by no means suppose that the transmutation hypothesis is proven or anything like it. But I would very strongly urge upon you that it is the logical development of uniformitarianism. So this is an idea that small successive variations, either in geological activity or in life, these small things over large periods of time can give you sort of a big changes. Things that before that people would have ascribed to the hand of God. So this is just the way it was received, and it was received based on its sort of philosophical appeal more than its scientific appeal. So then if you want to trace what were some of the highlights of biology, here is Johann Mendel. He was, I don't know if he was Austrian or he was Czech, but Mendel basically in the late 1800s did a lot of experiments. He was a monk and he was in the monastery. He was playing with growing various kinds of peas. And he became very, very systematic about what was happening when he crossed the pollen from one kind into the flower from the other, what kind of peas you get. And so there were attributes, for example, of whether the peas would be round or would they be wrinkled, whether they be yellow, whether they be green, whether their flowers would be in the middle of the plant or at the top of the plant. All these attributes could be tabulated, what happened, what were the parents like. And so he did this systematically over time, and at least some of these traits he realized fall into very distinct mathematical, statistical descriptions. And from that, he brilliantly deduced that heredity seems to be particular. In other words, a particle is given from the parent to the offspring that determines the offspring's traits. Later, people took that. By the way, he was actually not known until much later. People essentially discovered it much after his death, I believe. So he did all this pioneering work, but it was mostly not known at the time. Some great scientists later argued that this showed that Fisher, or R.A. Robert Fisher, showed that this showed that great variations are possible. Now, great variations is what Darwin would like. Darwin wants a lot of variation possible. Darwin is not wanting just limited variation, but a tremendous amount of variation. So Fisher, not Mendel, is saying that this Mendelian genetics shows that great variation is possible. And also, but also, Fisher, another thing that he did that was very significant scientifically is he kind of made the Darwinian theory a little more maybe realistic or a little more precise. He basically said that, when we say something is favored, it's favored because the offspring, you think of a population of some kind of creature. Now, among the population, there's a lot of variation, but those variations that are better suited to whatever is the current problem that needs to be solved. It's lack of water, too much heat, too much cold, whatever it is, that variation survives better, and therefore it's populations that are changing and how among the population the fittest survive is how Darwinian theory works. So that was great. And then as you move forward now into the early 1900s and now to the essentially mid 1900s, the tremendous discovery of there was an acid known to be residing in the nucleus of a cell, and it was called nucleic acid. But it was at this point in the 1950s that Watson and Crick, using X-ray crystallography, made the startling discovery of this fantastic double helical structure that just went on for great lengths. And so what they discovered is this double helix with these bases, what they call nucleotide bases, in the middle of it. This discovery had a lot to do with bringing more focus to the Darwinian theory and giving us essentially what we now call neo-Darwinism, the more fleshing out of the Darwinian theory. And so this stuff, you can think of it as the backbone, sort of what holds this medium together. And then the base pairs, turned out there were 4 of them: adenine, thymine, cytosine, and guanine. And these are bases, nucleotide bases. Now, each of these likes to connect to the other in a unique way. The T and the A, the C and the G, the A and the T is just like the T and an A the other way, and the T and the G. So if you have a T, you know the other side's going to be an A. So there's no variation possible. Once you have a T here, the other side's going to be an A. The point being, this side really is a mirror of this side. Whatever is going on here can tell you what should be here, and this becomes important later on. And so there was a lot of excitement about this. This was a magnificent discovery. The other big discovery that also in the 50s that made it look like Darwinism, every piece of it is now being completely proven, was the famous experiment of Stanley Miller under the guidance of I think it was his professor, Harold Urey. And this experiment was basically a kind of experiment where you make a certain mixture of gases that you would have said would probably be in the early Earth atmosphere. And then you zap it, where you put energy in there with electric spark. And then you just kind of wait and see if, as it kind of, this stuff basically drips down, if you can collect anything interesting in there. And sure enough, what they collected was a couple of kinds of amino acids. Now, the amino acids are super important biological compounds, as we will talk about in detail more. So by the late 1950s, it looks like we know how genetics works. It looks like we know now how how early life might have started. I think I need to tell you one other thing that was important about this, and this was what was called the sequence hypothesis. You know, the DNA, we said, has all these nucleotide bases, these bases that we have names, that we could just call them A, C, T, G. Well, George Gamow, who was a physicist, said, hey, look, this thing looks like these things are coding for something. This is a sequence of informational words, essentially. And the sequence hypothesis was a major idea that now it looks like the DNA may be a carrier of information. And so now this picture that I'm drawing is way past the time frame where George Gamow would have known anything about this at the time of the sequence hypothesis. But eventually it was made clear that this idea was completely right, that these latter steps, with all their names, G, C, U, A, these are just like groups of bits on a DVD. That just like those things basically tell you, code up, basically tell you what some information, like you want to watch Star Wars, you put the DVD in and it tells you what every pixel on your screen should be. Well, this thing now is just describing a huge amount of information. Now, I don't know if anybody knows how much storage you can get on a DVD. It's about 4.5 gigabytes. The DNA, the human DNA is about 3.2 gigabase pairs. And each base pair, I think you can think of it as being 2 bits. And so it's comparable. It's comparable to a DVD. Now, it was...
John, they've actually done that. They've taken pictures and put them on DNA and stored them in a test tube. Run read them back out again.
Wow, that's cool. So, total demonstration of the information carrying capacity of the DNA. Pretty amazing that they could be with the DNA. Now, we can do a little bit of game. Those of you who are computer science folks, if you have in each location in the DNA, you can have one of 4 possibilities, right? If you have 4 possibilities, what kind of how many, so you have, 4 chances to change the letters. 1 bit is 2, so each one of these nucleotide bases is like 2 bits. So you have 2 bits, and now you need to use the sequence of these 2-bit little pieces to describe any of the 20 amino acids that are found in all the proteins of our bodies. And it turns out if you work out the numbers that you need, if you also put in the additional constraints that needs to be because of the mirror nature of this, then you need 3 of them before you could describe uniquely one of the 20 amino acids. And so in fact, 3 nucleotide bases are called a codon. That means it codes for an amino acid. Now I keep mentioning amino acid, and I'm going to show you a video now, but the point of it is that the amino acid is the building block of the proteins, and proteins matter a huge lot. Let me see if I can get this guy started. So watch this.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a 2-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
One thing that should be pretty obvious is that up to now we were talking about, the DNA, and the DNA is obviously, there's a lot going on there, but when you actually get an idea of what the cell is doing, it is a galaxy. I mean, it is so much going on there that is staggering. Thank you very much. That's just absolutely amazing what's going on. Nope, that was last time. All right, so next I'm going to talk about, so up to this point, we could say that what we're seeing is a continual progression of detail and specification in the Darwinian picture. By the late 1950s, in 1959 in the University of Chicago, there was a celebration of 100 years of Darwin. And at that point, the Miller-Urey experiment was done, the double helix was discovered, and everybody thought, tomorrow the world, we'll get everything. This is clearly, there's nothing left, in terms of an argument. And then the 50s led to the 60s, and people, as the scientific progress went forward, a lot of new findings actually turned the story around. So I'm going to go into that. There is a multifaceted, deep story, and I'm going to now go into one part of the story really deep, and hopefully we'll come out of it not completely confused. So this is based on a book by Michael Behe called The Edge of Evolution, and I'm going to start it with a case study on malaria. So malaria is a disease that kills roughly 1 million people per year. And the origin of the word is malaria. It's like bad air. People thought that you were near these ponds, that the water was kind of thanks, but it kind of worked itself out.
She just touched the remote and it was
So this was, people thought it was from bad air, but it turns out it's about basically what's involved is a parasite, a very sophisticated parasite. It's single cell creature. It's called the Plasmodium falciparum is the name. See, I can say it, but I can almost sound like a biologist. And then this is the Anopheles mosquito that carries it. And what basically happens, and this is probably much bigger subject than I can go into, but I'll tell you the cartoon version of it. The cartoon version of it is that this parasite basically is injected into the bloodstream by a mosquito. When it goes into the bloodstream, the parasite knows how to find the liver in some way. Once it goes in the liver, it incubates there and basically grows into some appreciable numbers. From the liver, it comes out into the bloodstream, where every one of these instances of parasites finds a red blood cell and infests the blood cell. It then goes and basically starts eating the blood cell from the inside, the hemoglobin, and pulling out the amino acids from the hemoglobin. Now, hemoglobin is a protein. It's made out of hundreds of amino acids, basically. And the amino acids are like the Lego blocks of protein, and it uses the Legos to build another one of itself. And then once the poor little red blood cell is all destroyed and all consumed from the inside, these guys burst out, each 20 of them, and each one, each one basically goes and infests another red blood cell, and it continues at factors of 20 each step, so that it exponentially grows to hundreds of millions in one body.
Trillion.
Actually, yeah, I was going to say billion. I didn't know it was
Trillion.
In one body? Oh, that's right. Yeah, that's right. You're right. Yeah, that's right. Right. Thank you. That's great. So basically, it just totally overwhelms your body, and it's all over the place. And now what are the problems you're having when you have this? Well, the first problem you're having is your red blood cells are all being used up. So now you've got weakness because the red blood cells are involved in bringing oxygen to your cells. And so you're anemic. That's at least one major problem. And then eventually, that's all over your blood now. So then this mosquito comes, and next time it bites you, it takes some with it to infect other people. And that's how 1 million people die every year. So that is the bad news. But then what happened was that in the 17th century, people brought a powdered form of the bark of a tree, the cinchona tree, from the Andes to Europeans who were in Africa. I don't know the story of how it made it all the way to Africa, but these Europeans who were catching malaria and getting very sick took it, and they got better. Now, why weren't the Africans benefiting from it? It turned out in Africa there was a mutation that was the sickle cell mutation that was conferring an advantage against this parasite to the Africans. The sickle cell hemoglobin is a mutation of ordinary hemoglobin. It is a sort of a damaging of ordinary hemoglobin. But it turns out to be a kind of damage to the hemoglobin that when this nasty bug is here, it gives you a little bit of an advantage. So this is part of the story we're going to talk about, how this kind of thing is actually a bunch of damage that it's fighting against other damage. So eventually the active ingredient was found to be the chemical quinine, and then in the 1930s, the German pharmaceutical companies, I think it was right before the World War, they basically discovered they were going to manufacture quinine. And suddenly, basically, quinine was able to really stop malaria. I mean, it just, I'm going to talk about, I think I have a slide on how quinine works. It's very interesting, actually. And then after World War II, the Americans were able to figure out, the Germans had these quinine, they wanted to make it, but they kind of figured out there's a simpler shortcut to the quinine called chloroquine, much easier, much cheaper to make, and it's just as good as quinine. And they used chloroquine abundantly, and malaria was almost eradicated. Malaria was eradicated then, but it's not eradicated now. Malaria is a serious killer now, but in the 1950s, it looked fantastic. It looks like malaria was a thing of the past. Okay, so what happened? Well, okay, before I tell you what happened, let me tell you about basically why did this drug work, because this is kind of interesting too. So the malaria parasite feeds on hemoglobin, as we mentioned before, right? And so it breaks up its amino acids and kind of uses it to replicate itself. Now, a hemoglobin is made out of, kind of roughly out of 4 myoglobin proteins, each of which has this little piece in the middle called the heme group. And the heme group, basically what the heme structure, whatever we call that, polymer, now biology needs to help me. But the heme has iron in the middle, and that iron can attach to an oxygen. So what the heme group does, and especially the way the machine that the hemoglobin is, there is, almost think of springs and latches and things that hold and then that let go. So hemoglobin is built in a way that when it's in an oxygen-rich place, it can grab oxygen that almost nothing else likes to touch. Think of oxygen as like a piece of live wire that's at 1,000 volts, that you see it there, it looks innocent, but don't you touch it. Okay, and in biological systems, oxygen is like that. It's very valuable, but it's also pretty dangerous, pretty nasty. And the iron also acts like that. It has this feature. So that the heme is actually, when this bug comes in and consumes the hemoglobin, it hates the heme part. And it kind of like wants to make that into a ball and get rid of it as waste product. What the chloroquine does, or quinine, or either of those, what they do is they make it hard for this part of this, the parasite, the cell, to create this hemosome, which is this bunch of the hemes all bundled up and ready to be thrown into as waste. People are not sure how it works. They think that maybe there is essentially a pump at the surface of the food vacuole, that there is a protein that is working there to kind of let the good stuff in and bad stuff, like this heme bundle, out. And when you put the chloroquine in, it somehow, the pump doesn't work right, and so it can't get rid of the heme. And when it can't get rid of the heme, a wonderful thing happens to this nasty parasite. It dies essentially in its own waste because this heme is all over and it can't do anything about it, and it basically destroys itself. So that's how chloroquine works. And so what was involved? What was involved was this one protein, PFCRT. Now it sounds really fancy, but if you actually find out what it means, Plasmodium falciparum is the name of this parasite. So PF is just that. And this is chloroquine resistance trait. Normally this is what happens. Normally what happens is that this protein is part of a pump that when everything's working properly for this malaria parasite, it gets rid of this toxic stuff. But the chloroquine was getting what was basically not letting it do that, and the PFCRT is involved in all of this, and I'll mention that in a moment. So this was all interesting, and chloroquine was used, and it was inhibiting, it was causing this parasite to die. But starting in the late 1950s, suddenly the first reports came out that chloroquine had stopped working. So it first started in, I think, Thailand, and eventually all of the world by the late 90s. Chloroquine is suddenly a failure. So what happened? So the question is, what happened? And so apparently what happened is that Darwinian evolution took place. And the evolution essentially enabled this parasite to become resistant to the chloroquine. And in fact, what in fact was found to have happened, okay, is that
Iran stayed resistant.
What's that?
Iran was spared from this chloroquine.
Oh, well, yeah, that's why I'm here. So let me go back here. And basically then, this pump was, the chloroquine, I think, was affecting this pump's working, and therefore the pump wasn't able to properly get rid of this stuff. But now it figured out a way so that it didn't care that the chloroquine was there, and the pump kept working correctly. That was what chloroquine was doing. Now, how was it doing? Let's go a little bit further. And this is now a pretty good story about how an organism becomes resistant. Now, this is Darwin theory, Darwin mechanism in action. So it said during cell division, okay, there can be sort of errors when the DNA is replicated. So you have a cell, and the DNA replicates, and eventually you have two daughter cells. Of course, this is a cartoon, and a vast amount goes here that I don't know, and I'm not going to be able to explain. But the great point is that DNA that had the codons for the amino acids that make up the proteins, such as PFCRT, that DNA essentially gets replicated in this step. When it gets replicated, the fantastic machinery of the cell doesn't let it make mistakes. There's error checking, and there is a really great process for taking billions of codons and reading them and not coming out, wrong. Except maybe 1 every 10 to the 8th. So 1 in 100 million codons will get messed up. Now in this creature, there are its DNA is about 10 to the 8th codons, or 10 to the 8th, codons. And so, approximately 1 codon can get damaged. Changed. And so when a codon changes, by the way, what changes really is, and that was in that video, where each codon essentially codes for an amino acid. So imagine, in the DNA, you say, I want this amino acid, then I want this one, then I want this one, then I want this one, stop. And then now make this one, this one, this one. That stop and the start essentially give you an amino acid sequence. And there's another part of the cell that essentially enables it to fold up and become some fancy shape, like it should become a saw or a hammer or whatever it needs to be, that protein needs to be. The protein's shape is determined by the proteins and the way they fold. Okay, by the amino acids and the way the whole structure folds. When you change an amino acid, a lot goes bad. It turns out that, this is a vast topic, and books are written about this. You cannot change these amino acids without dramatically changing the protein and its function. So anyway, so the error rate is very small, and then there's error correction that makes it even smaller, but when it does happen, like I said, you end up with the wrong amino acid. So you've ordered the wrong amino acid for this part of the protein, and so it can affect the way the thing folds, the way it can bind to other proteins, and so those things all go bad. And all of these cause damage of function. That's a very important point. So that in every instance known to us, to science, that this process of mutation breaks something. It never fixes something. So occasionally, there's an advantage due to the door the thread is coming in. Advantage due to the door essentially breaking. In other words, imagine that the problem you have right now is chloroquine, and chloroquine is essentially damaging your protein. If you now damage your protein in a different way, further, you can make it so the chloroquine doesn't do any worse damage to it. People have likened this to what used to happen a lot in Europe during World War II, where your village or your town is being attacked by the enemy, and you've got a bridge, and they're about to go over the bridge, like in this one in Belgium. The people blew up their own bridge. By blowing up their bridge, they made it impossible for the tanks to roll in from the other side, at least right away. And that gave them essentially a little advantage against the threat that was coming. At the end of the day, though, they lost their bridge. And evolution works like that. Darwinian evolution essentially takes advantage of the random mutation to break things. And a lot of those breakings are not good for you. In fact, none of them are good for you, except that sometimes accidentally something breaks that was the doorway through which you were under pressure from something. And so you end up breaking the right thing, like breaking the bridge. Like an intruder is coming to your house, and you know it, so you go into a room, you close the door, you jam something into the lock so they can't come in. You survive that evening, but your door just got damaged. It's okay, you can go buy another door. But the point is that's how Darwinian processes work. They break things because that's all random mutations can do. That the molecular machines and the molecular, that the proteins are super, super custom. They are fantastically intricate. And the only thing you can do is blow up one, and randomly, one of them that you blew up is the right kind to grow up for right now. But if you then took this organism and put it back into its parent population without the threat of this problem that it was solving, it does not have a selective advantage. It goes away. It's not better than it used to be. So that's the story of this. And so,
Hey, John, are you
yeah.
saying if we all stopped taking chloroquinone, that the malaria parasite would stop being resistant to it?
You know, in fact, you can start taking this to, yeah, I think so. The problem is that, you know, millions are going to be dying, but then you actually, if you take a step back from that, you could do this. The other thing you can do, and people do immunologists, I don't know, is you can use cocktails of drugs. Think of it as here's the enemy coming in, and here's all the different ways it can come in, and you think of a line as a barrier, but the barrier essentially works everywhere except one place. It doesn't work. If you then use another drug that works in this other place, then you have essentially, you know, you multiply the probabilities of success or the failure. So anyway, so a little more detail on the numbers of this is this. So there was a study done in Thailand that estimated, they looked back and said, what happened with this malaria thing, with the chloroquine? And they said, well, look, it took approximately, they could see how many trillion was inside an average person. And then they said it took, you know, what was the population of people who actually contracted this? And, yeah, and so roughly numbers like this, and they said then approximately this many years later, you can expect, and it was about a decade, they said that you can expect that the mutation should have occurred. And so then the 10 to the minus 20 became a number that was, or 10 to the 20 parasites became kind of the interesting number. So then smart people like Behe basically said, well, if that's the case, that means, remember we had a number that we were carrying in our back pocket that was 10 to the minus 8 correction, you know, essentially the likelihood that a mutation can survive error checking is 10 to the minus 8, 10 to the minus 9, 10 to the minus 10. And so people guessed that there might be 2 amino acid changes, that because 10 to the minus 10 times 10 to the minus 10 is 10 to the minus 20. 2 amino acid changes that might be involved in this. Now, as I say this is in Darwinian circles a bombshell, because I just basically destroyed Darwinism in saying this. Why did I destroy it? Because here's the problem. If you're talking about malaria, these organisms that are, you know, 1 trillion of them are in one person, and millions of these could catch this within some number of a few years, the process of getting to 10 to the 20 of these is not that hard. You can get to 10 to the 20 within a few years of these parasites. You have 10 to the 20 opportunities to get the lucky 2 mutations, the very 2 that you need. Now, what have you achieved in that 10 to the 20? You've changed 2 amino acids of 1 protein to encounter a problem. You haven't done anything grand. All you've done is make a tiny change in 1 protein to damage it. Okay, here's the point. The point is that how long do you think it takes for humans to reproduce 10 to the 20 people? Think about that. Or how many, in fact, how long do you think it would take? You know, it's one thing for bacteria to develop, E. coli, to get 10 to the 20 back, you can do that in 10, 15 years. But what about little mice? How long does it take to do mice? Maybe 100 million years?
Well, if they're in our attic, about 1 year.
Yeah, that's right. So, and this is a very fundamental problem. And so there was a lot of controversy when this result came out. And this paper was sort of the final vindication that in fact it needs 2 mutations. And that 2 mutation thing was really a big deal. Because everybody said it must be 1, because 1 is easy. If you get 1, and then you can get a second.
Paper came out after Michael B. put his neck out and said it would be 2 mutations.
That's right, that's right. People used to argue with him, and then the paper came out and vindicated him. So this is a very big deal, because for anything bigger than at this size or bigger, 10 to the 20 is a formidable number. Basically, you can't get that. And remember, when you get 10 to the 20, what have you achieved? 2 amino acids in 1 protein. And you think about hundreds of millions of years and what evolution is supposed to have achieved in hundreds of millions of years. I mean, the Earth is only, 3.5 billion years since when it was a molten body of rock, right? So you have, 3.5 billion years, and in 3.5 billion years, it takes, a good fraction of a billion years to change a protein. And you think the difference between us and the supposed common ancestor is 1 protein? Obviously not. So there's a serious, serious problem here. Further difficulty comes like this, that a lot of organelles, these are little machines inside the cell, are made out of not 1 protein, but multiple proteins that are fitting each other, like this protein and this protein, and they fit each other like a glove. And for them to fit each other, their shapes have to be the same, but also the electrical nature of these, whether the edges are hydrophilic or hydrophobic, whether they're plus and minus versus plus and plus, there's 1,000 ways this can go wrong. So when these things have to fit, they have to fit perfectly. And so these are called binding sites. So when you are trying to make organelles out of multiple proteins, you have to create matching binding sites. And a question that can be asked is, when we are looking at cells that we can reproduce rapidly, like E. coli, that we can reproduce in giant numbers, how many new binding sites have we seen after billions and trillions of these things being regenerated? And the rate is basically 1 new one has been seen. Actually, it's been So the probability has been set as not better than 10 to the minus 8, almost the same as that other number, the mutation number. So that means if you've got a protein that has a day job, and now you're going to say evolution, by random mutation, it's going to have a new function. Like a light-sensitive cell is going to now become some part of a compound eye or whatever. So it's going to change its function a little bit. And you say, well, okay, then part of that is because, it's usually not 1 protein, it's multiple proteins. Like, for example, hemoglobin is 4 little subproteins, myoglobins roughly, that kind of make up hemoglobin. These guys are fitting together the right way, and they make up hemoglobin. So the point is this. The point is that if you need 2 proteins to bind together correct in a new way, you have to have 10 to the 8th reproductions, like 100 million, okay? Reproductions of that creature before this 1 protein accidentally you get 1 kind of favorable change. Now, there's a lot of other issues there, but in fact, most organelles are not 1 protein with 1 protein. What about a 3rd protein to these 2? What about 50 proteins, which is very possible? What about 1,000? So you can get very, very large number of binding sites. If you need n proteins to bind together, the probability is 10 to the minus 8 to the nth power. So just like, you have 3 or 4, you run out of probabilistic resources. There's no more, you cannot just random evolutionary-wise get to these kinds of numbers. And that's what protein binding site. The protein binding site is the analog of this problem, but it's basically another aspect. So what do we take from this part of the talk is that number 1 is that the Darwinian process does work. The Darwinian process does work. But what does it work? It works at the level of breaking simple proteins to overcome small problems. But it breaks things. It doesn't create things. And so that you have this irony, because at the same thing, You remember Darwin said there was natural selection acting on random mutations. What this story tells us is that in fact was brilliant. It is true. Natural selections acting on random mutations work, those two things. But they only work to confer on creatures the ability to not all be annihilated by one parasite or by one, thing. The variation causes the overall species to dodge bullets. It's sort of almost like protection built in to the world we live in, that these variations occur and the whole species doesn't go away. But the moment you ask for more, the moment you ask for anything beyond these very trivial changes to like maybe a couple amino acids and a protein, you're asking too much. It just doesn't work. And in fact, those same things, random mutations and natural selection, become your enemy in going further, because natural selection stops working properly and random mutations stop working properly, or doing anything more interesting. So this is basically, I would call this the mechanism challenge to evolution. But the mechanism isn't working. The mechanism works well for micro processes, and it immediately fails right past species. So when Darwin goes and sees the Galapagos finches, those Galapagos finches were changing because of mutation. But those are just variations within a species. You just can't go past them. These processes don't let you go past them, and you're kind of stuck there. So what are there are other fundamental problems with the Darwinian process, and this is the part where I'm going to just hop over very, very big topics in just one slide or two. One of them is also by Michael Behe, is irreducible complexity. It's the notion that many of these objects that are at play at the cellular level are really molecular machines, and the molecular machines are irreducibly complex. And what is irreducibly complex? Think of a mouse trap. A mouse trap that you buy, a nice cheap mouse trap, has been built so it catches mice. It's been built so it's cheap for the maker. So the maker is not going to embellish this mouse trap because it's going to cut into their profit. So it's got just the minimum you need. It's got it's got this.
Parts.
What's that?
5 parts.
Yeah, 5 parts. And in fact, I could use help. What do you call this one? The hammer, the catch. I think that's the catch. Yeah. I don't know what that is. The yellow thing.
The cheese.
And this base. And the point is that, and B makes this point, that this thing is irreducibly complex. You take one of these, and the challenge is take any one of these, you can choose one part to take out, and see if you can catch most with it without that, once you take one part. It has been reduced to the bare minimum it can be. You can have a little LED light here, and you can have a little play songs for the mouse. Those are more, then it would be reducibly complex. But once you take the LED and the little song singer thing out, this is the minimum you need. And the point is that there are molecular machines in the cell, for example, the bacterial flagellum, that are fantastically complex. When you look at this, when the first time people looked at this under a microscope, oh, isn't that cute? It's got this little swinging thing. It looks like it's just a little smooth thing. Darwin himself thought of the cell as just this gunky thing that was very simple. Sometimes it's easy to look at something, and your first reaction is that it's simple. When you look closer at just the tail end of this is what you've got. It's an outboard motor with a stator, which is basically sort of like a permanent magnet, and then it's got a rotor, it's got a drive shaft, it's got bushings, and then there's a corresponding version here. But if you were to build an electric outboard motor, you would need all these parts. It's irreducibly complex.
It's also self-assembling.
And it's self-assembled. That wasn't enough. That is right. That is right. So the key is that irreducible complexity is a very big topic. This is a trivial example, even a much bigger example. I mean, it's not trivial at all, but if you actually go into the account of blood clotting and what blood clotting is about, it is staggering, the flow chart that you have to draw. Because there are proteins that allow the blood to clot. But if you don't watch out, your whole blood clots and you catch a stroke. So you need something to actually regulate that. But then if you do it too much, you bleed to death. So something has to regulate that. And it goes on like 20, 30 levels, and there's all these diagrams because everything is controlling each other, and it's just a super complicated thing. So this is from Michael Behe himself. He picked out this Calvin and Hobbes cartoon to say that sometimes when you look at something the first time, you can have a simplified idea. Calvin here thinks when he's seen airplanes, and it just fits in a cardboard box, it's got little things like the wings. Oh, you know, you can imagine he's got most of an airplane. But when you look closer at the airplane, it's a little hard. So now I'm going to leave the vast topic of irreducible complexity, go to the vast topic of the origin of information. So remember that we said DNA holds 3.2, that's incorrect, 3.2 billion base pairs. I forgot billion. Or over a billion 2-bit codons, okay? Where does that information come from? Information invariably comes from a mind. Random processes invariably destroy information. So the point is this, that when you look at the DNA and you look at this vast amount of information that's there, every process we know in nature destroys information. And the only process we know for creating specified information is an intelligence. Now this has been known since the 60s as a fundamental problem with the neo-Darwinian synthesis. But what was in vogue when I was a postdoc in the 1990s was people were throwing around this phrase, self-organizational systems. And so the idea there was that you know, these molecules that you're allowing to put in, once you put in one base pair, what if that one base pair kind of selects what goes above it? And so you just have to get one base pair together, and then the thing rebuilds itself up all the way. There's a very fundamental mathematical problem with that. There's a physics problem with that, because the bonds are weak between these ladder rungs, actually. So there's not that much influence. But if you then do the null hypothesis, say, okay, I'll grant you. Let's say it is that way. Now imagine, liken this to a letter. Let's say that you're going to college, like some people might be, and they write letters to their parents, back when they used to write letters. And they said, it starts with, "Dear Mom," and then, you know, "How are you doing this week? The weather is nice here, how are you?" And if every time you get the letter, it starts with, "Dear Mom, how are you doing this week? The weather is good, blah, 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 blah." And it's actually the same words, that in fact, you know, your child gets so used to this that as soon as they start picking the letter, they just write, "Dear Mom," the next sentence is the same thing. There's no more information in it. The next time the letter comes, you just go, okay, then at least this child is alive. So there's one bit of information in it. The rest of it I can throw away. There's nothing new in it. And to the same extent, if the "Dear Mom" tells you the rest of the letter, there's nothing left there to describe, because the first molecule defined everything. And so the information capacity, carrying capacity, goes to 0 as the probability for the sequence goes to unity. So the information carrying capacity, this is the Shannon information, is essentially a measure just like entropy. It has to be very low probability before it can carry information. So this is called the origin of information problem. There is a ton of information, and the problem is that you have only two choices if you want a materialistic picture. You have randomness, and you have basically law. I was just mentioning law. In other words, physical laws, you know, electrical interactions determine what the whole thing is going to do, but if it's law, then there's no information carrying capacity. So then you need randomness. And the problem with randomness, as I challenge any computer science student, write your next short piece of code in Python or R or whatever you're going to write it in. Write a little program that writes the program randomly. See if you can manage it. Just throw random bits, and then feed the binary into their machine and see if the binary runs on the machine. And then try to do that. How many trillions of times would you like to try to do that? See if it ever does something correct. The truth is that every way that you look at it, information cannot be. So people used to say that, you know, the universe is, we need the universe to be very old, because if it's an old universe, then there's no time for evolution. The problem is old, young, whatever, it doesn't change anything. Time is not the healer of evolution's information problem. Time actually destroys information. Time and thermodynamic effects. So randomness is a definition, and then I'll tell you that it's a stupid definition. Okay? And I suggest organization as a state of low entropy. And I say it's stupid because it's correct, but it's correct in the most trivial way. Organization is far more than a state of low entropy. It's like saying to be, I don't know, what's a good example? It's to be a television set is to be boxy. Yeah, televisions are usually boxy. Or used to be, except now they're kind of plainer. But there's a lot more to a TV than a box. And so here's the point. An organized system is composed of components which have function. So, and that can work in a software paradigm, it can work in a hardware paradigm. You know, you write a constellation, I mean a package, and the package has all these functions. You call your main calls these some functions, these call their own functions. Each has inputs and outputs, and they all expect it to be correct. And if you're going to pass this out to the world, you're going to have to unit test this thing so that it actually does all that stuff correct. So those are components, and they have function. And if you want to look at a system from the outside and say, gee, I wonder what that does, you pull it, and the state of the system after you pull it, minus the state of the system before, was the function. In other words, if you start going, you go, ah, I see. That one was a spark plug, you know. And so you can formally, mathematically define what function is. Function is the state of the collective system with this part not there, you know, minus the, or the sign, minus the system with the part there. And you can, you know, people have, there's a very hard to read book by a man who's now passed away, the name of Robert Rosen, who basically defines this way. And he says, if this is true, and it is true, then the first thing that the typical materialist biologist does, which is to essentially, you know, basically put the organism in a blender to basically do assay on the various compounds, is to first destroy the biology, and then basically goes and looks for the chemistry, which is useless. At that point, the biology is gone. Biology was at this level. And you destroy, and the normal Darwinian paradigm for doing science actually destroys the biology first. So another way to say it. So how now science? So how do we do science? So the physicist, one way is you can look at it as a physicist. You think everything is reducible to physics. And so you see a baby and you start going, well, okay, there's atoms, and the atoms make up, you know, biomolecules and DNA, cells, you know, and then somehow these things evolve and we got the baby, okay? The engineer looks at it backwards. They look top down. You open this you know, box, and you see this thing, you know, let's say that you get this, you know, drone from a rival country. Actually, I have a nice slide that John made for me. So in the waning years of the war, the most fancy airplane in the world, fighting warplane in the world, was the B-29 bomber, very most sophisticated airplane. A bunch of these crashed in the Soviet Union, okay? And after the war, the Soviets showcased their brand new Tu-4, Tupolev-4, that was their creation. And if you look at the two pictures, you see that they essentially copied the crashed bombers. And so what were they doing? They were doing it the way we say science, perhaps you would want to be doing it, which is that you go backwards, you open this airplane and you go, so what are the systems in this plane? It's got the engines are made this way, they're rotary, they've got this much power, blah, 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 blah. You don't say, well, let me first time the engine start, I'm going to start with one bolt and figure out how this bolt works. No, you're going to draw the thing from outside in, and eventually worry about what, you know, how much pressure or stress that bolt requires. You want to now first know, you know, what is the wingspan, how much thrust, how much, you know, lift, how much drag. You know, that's how you're going to come into it, from outside in, because you're now thinking about systems and architecture. You're not thinking about the particular things. And so there's a whole different program of science when you think about it this way. So we talked about that. And so here's a cool quote that I kind of put in last minute from Doug Axe. It says, Darwin's idea that life wanders from one variation to the next, never committing, always yielding to the blind forces of natural selection, is plainly incompatible with the idea that the physical forms of life are expressions of something deeper, something immovable, something perfect. And he makes this nice story about, You know, when you look at a salmon and you look at an orca, you know, the salmon is all salmon and the orca is all orca. There are so many things about the orca that make the orca what it is and what about the salmon. The other thing is that the Darwinian process, I like this plot so much, I just put it in. I don't even think it goes with the code very much. This is essentially an optimization aspect of the problem. Now, those of you who are in computer science or math, sometimes you have to do numerical solutions to find a minimum, like a local or global minimum. You're trying to do it like a least squares minimum. Well, least squares is easy, but you have to do this like this nonlinear problem where you have to like try various answers and you want the optimum answer. Darwin essentially thought fitness landscape would look like this. You're a creature, you have certain morphology, you have certain attributes, but when you vary, your children vary in the different directions. But one direction always leads to getting better and better and better and better. And you will keep climbing this mount until you get to the, to the perfection eventually. It'll take billions of years, but that's okay. What people are finding is that in many cases that you actually study, the fitness landscapes look like this. You are on this little island, let's say this little island here, and your little progenitors, they make a little step and uh-oh, that got bad, they come back. They go this way, oh, that's bad too, and they're blind. They don't see there's a bigger peak over here. Remember, Darwin is the blind watchmaker. It is blind. Yeah, Dawkins was talking about the blindness. Blindness means that you can't see that. You're just stuck here. And every step you make tells you, oh, that wasn't good, and you're stuck. And so the fitness landscape is another fundamental problem of the Darwinian picture. So we are in a world that is transcendently designed, and it has attributes that include beauty, laws of physics. When you look at the word engineer, it says skillfully and deliberately arranged. So laws of physics, they don't care. That's what their purpose is. They're laws of physics. They're laws. They work. Designers, on the other hand, care about successful function. Artists care about beauty. And both attributes are there in the living things. Beauty and function are there in huge proportions. So, and so what we're seeing that the living things are engineered and they're artfully built.